So it's week 8 discussion. Did you guys start doing the homework already? I don't think it was too bad. Some things to talk about for the homework. This whole homework revolves around one type of cancer called acute lymphoblastic leukemia, or all. The all doesn't mean we're working on all types of cancers, it's only this type of cancer. There's 2 subtypes that we downloaded for this experiment. There's the BCR/ABL, which the ABL is another way of saying ALL. And then there's the ALL1 and AF4. So yeah, there's 2 subtypes. Just because these samples were taken on like a certain date, there's more information known about them. So specifically what type of cancer it was, or if it went in remission. There's also other types of information, like the gender, the age, the date, if the cancer came back, when was their last treatment, all this stuff's kind of called the metadata, just describing your data. So that's what the homework's about. They use heat maps a ton in the homework. I'll talk about that in a second. Just some hints for the homework. The first one says, or not the first one, but one of the questions says list a couple pathways. Don't just list the same pathway. List like kind of 5 different ones related to it. What do I mean by that? Some of these pathways are pretty much the same thing. It's kind of hard to see on the screen, but so one of these is called positive regulation of T-cell activation, and then another one's called positive regulation of lymphocyte activation, and then there's a third one called regulation of lymphocyte activation. Well, if you think lymphocyte activation is relevant, don't just list positive regulation and then list regulation of lymphocyte. Pick 5 different pathways for this one. I'm sure you could find 5 different ones on this list just talking about the same thing, but make sure they're different. That's my hint for that one. So the other thing they ask you about is kind of looking at clustering of heat maps. So, I mean, the non-scientific way to do it is just to kind of squint, and if you see a big block of color, that's your clustering of the heat map. What do they mean by anomalous? Well, there's, when you squint and you see like a little blob of green and a red, or a little blob of red and a green thing, that's what they're asking for like anomalous. Another anomalous is if 2 large blocks don't sit next to each other even though they look like they do, or they should. That's another thing you can talk about. It's like when I was in Florida, Gainesville was a Democrat, but everyone else in Florida was a Republican, so there's like a little dot of blue inside the big red state. So those are like the anomalous spots of the heat maps. Yeah, so I mean, that was pretty much the hard stuff for this homework. I don't think it was too bad. It uses the R programming language, which it should be straightforward if you're just pasting stuff in, but if there's any problems with it, you guys haven't had any experience debugging it, so that could be rough. But other than that, it's kind of just pasting stuff in, and you're really asked about the biology for this stuff. So this might be pretty brief, but if you guys want to ask me questions about the homework, I've got it here. Ask away. Yes?
This part where I think it was you want to look at the regulated point.
Yeah.
Or the top half. I think there's a top regulated gene.
Okay.
And you know, you define like this gene as something like reset or whatever. When you print it out, you get like sort of like spit something out.
Yeah.
Yeah. And the question says like, you know, how many genes are there?
Yep.
And when it spits out something, it says, you know, there's X amount of features.
Yep.
And there's 47 samples. So is that X number of features? Is that the number of genes?
Yeah. The reason they're called features is because the tool doesn't know if you're using a microarray or like sequencing or something. So they don't really know what you give it. They just know you give it a certain number of things. So they call them features. But those, they could also call them like probes, or they could call them genes, or it depends really on what type of microarray you're using. But the features are the x-axis, like what you're looking at, or whichever axis it is. And the summary tells you how many features you have. But yeah, I think you figured it out. Yeah.
For this is like 5 pathways that seem relevant given biology and LL. You said not to have them be too similar. I think a lot of these are actually very like interrelated. I guess I'm wondering how similar is this?
I mean, similar in just like the language. If one says like regulation of X and one says like regulation of positive X or something. Yeah, if there's only a one-word difference between them, chances are the biology of those two pathways is similar. It's just how they computed them to get the terms. Yeah, the idea is to list similar things biologically, but not just list the same pathway 5 times just because it's called like T lymphocytes or whatever. Whatever. Yes.
How do we know which one is like BCR versus like ALL1 or something?
Part of the code in, I think the last bit, is when you actually get into that.
Because it was like
So it says here on part D of question 4, it says if the type is ALL1/AF4, its color whatever FF00 is, and then if it's not, then it's the other one. And it doesn't really matter specifically which one is which. I think FF is red. So if it's a red box on the top of the heat map, that means it's the ALL thing. So I think this is the heat map that has it. So the red on the top column is what you're looking for when you look for your cluster. Did that clear it up?
yeah, so we're just gonna have to try to match it to the letter right there?
The big idea is that if the column, if this block is red, then it's ALL4 or whatever that one was called. And if it's blue, then it's the other subtype of cancer.
So what are we like clustering in like the first heat map that we make? Like
The first heat map is, I think it's described, but the equation for it is, give me the heat map of all the expression of this, the expression set, and then genes just number 1 through 50. So it's not really looking at anything specific. It's taking all the 20,000 or whatever genes in human cells, and it's only giving the top 50. And if you just took 50 random cells from or 50 random genes from an expression set, I wouldn't expect them to be correlated too much. But it's really just giving like an example first heat map kind of.
okay, because then it asks us a question about like how reliable is it, but it doesn't really look like anything clustered at all. So I'm confused about what that question is. It's like reliable about what? Like what information is it really correlating together?
Did they ask that when they tell you to look at the remission stuff too? Or they just asked it originally?
I think they ask you every time you repeat that.
Well, yeah. Then you start to do some like you like update it, and then that question like makes sense.
Yeah.
But
I mean, the first one is just asking when you squint at it, do you see blocks of color? That's the answer. So. Yeah, I mean, for all of these, if you say it doesn't look like it's clustered and you explain why, then that's the answer. Whoops. Yeah, so the first one pretty much just says, do you see any clustering going on here? That's up for you to decide. Okay, well, like I said, every time you can leave whenever. I'll be around for the next 40 minutes or so if you want to just go through it. I think the biggest problem, if you do have any problems with this assignment, is just going to be if something goes wrong and you have to figure out why it goes wrong. But assuming everything goes right, this is really just kind of exploring gene expression for the first time. So, okay, I'll be around.